Good morning.
Good morning.
Happy Friday. Yes, please type your first name or your nickname for me into chat. Thank you very much. Turn this on for us. A little more light. Good to see everybody.
Good morning, Professor.
Good morning. Good to see everybody today. We're just at about 11:20, so I'm going to go ahead and just go through just a reminder, as usual, about our schedule. Today in lab, we're going to be finishing up our discussion of special media. And if you remember the last time we were together, we sort of set up an experiment to see if we can identify two organisms that are two unknown species of bacteria that are mixed together. And we're going to use a couple types of special media to grow them and see if we can figure out what their identity is. So to begin things today, let's just take a quick look at our schedule, our upcoming schedule, what's coming up for us next week and so on. I'm going to go ahead and share my screen with you. If I can get it to share. Not quite. Hold on, we'll try again. All right, this looks good. There, hopefully on your screen now, you're looking at our course content page in Blackboard. So I'm just scrolling down. And here we are in week 5, last day of week 5 for us. And you'll see that there's a new content folder now up here called Pathogen Assignment Materials. I'll talk about that in just a minute. I'm going to click on next week's content folder so we can look at our to-do list together for next week. You have 2 lecture topics next week, typical week for us. One of them is microbial growth, and the other one is control of microbial growth. And of course, each one of those lecture topics has a quiz associated with it. We'll be meeting for lab as usual. We're going to talk about 2 subjects. One is oxygen and the effects of oxygen on different types of bacteria as they grow in culture. And then the second topic next week is going to be how we use ultraviolet light to control the growth of microbes in our environment. And of course, you'll have a homework assignment associated with those things next week as well. So that's up for next week. But I want to take a minute now and click on this new content folder, which is called Pathogen Assignment Materials. If you look into our syllabus and schedule, you'll note that there's something in there in the assignments, special assignments column called the pathogen table. This is not really so much about a table, it used to be, but this is more of a writing assignment that you'll be doing. I'm going to go ahead and click on that content folder, and you'll see there are 3 items in here. One is the instruction sheet, and then there are 2 links for submitting things to me for grading. You're going to be doing this writing exercise, and you're going to submit it in 2 different documents. You're going to first submit it as a first draft towards the end of March, and then I'm going to make some comments for you to guide your revision, and then you'll resubmit it as a final draft later in April. So let's take a look together at what this assignment is about. Hold on just a sec while I clean up my screen here. Let's see if I can share this with you correctly. there. So what you should be seeing on the front of your screen now is a document that says pathogen project up at the top. And you'll see there's a first draft and a final draft, so that this assignment is worth 50 points total. So it's a nice big chunk of points that you can earn. We talk a lot about what pathogens are like in this course, but we don't have much time to talk about individual types of pathogens, and the diseases they cause and so on. So that's what this assignment is about. It's going to give you an opportunity to do a little research into a pathogen and answer some questions about that pathogen. So there's all kinds of instructions here for you about how many references you should use. I've given you a list of reputable websites that you can go to find information as you answer these questions. And again, I've given you some very specific instructions about how to write your answers. So basically, you're going to be writing answers to a series of questions about a pathogen, and you're going to write them in paragraph form, in complete sentences with correct spelling and grammar and all that good stuff. But you get to choose a set of questions to answer. And you can see there are 4 groups of questions about 4 different pathogens. For example, the first set of questions is about the pathogen that causes tuberculosis. And the second group of questions is about the pathogen that causes cholera. And the third group of questions is about the Zika virus. And the fourth group of questions is about Staphylococcus aureus and food poisoning. So you're going to choose one group of questions to answer. And then, like I said, you're going to do some research and write up the answers to these questions and submit them to me. So it's pretty straightforward. It's not the same as writing, a research paper, because I have specifically asked you a set of questions that I want you to answer. But it is a writing assignment, and that's why I am asking you to write in complete sentences with correct spelling and put your answers into paragraph form and all that sort of thing. All of us always need to practice our writing skills. It doesn't matter what job you hope to have in your professional life. You will be doing some writing, sometimes more, sometimes less, but everybody has to do at least a little writing in their professional life. It's always worthwhile to stay in good practice. So that's part of what this assignment is about. And again, the other big piece of the assignment is learning a little bit about one of these very common pathogens by answering those questions. So that the first draft for that assignment, again, is due on March 24th by midnight. So you'll want to start working on that assignment, as soon as you're able, and start looking at the resource materials. And a good place to start is in that list of websites that I provide for you. So I'm going to let everybody take a look at that assignment after class sometime. And if you have any questions about it, you can either bring them into lab next week, or you can send me a message and we'll talk about it together over email. All right. So the last time we were together, we were talking about special media. And what you should be looking at now is the slide from last time that says today's exercise. And the bugs we're using, the bacteria we're using. And there's information on this slide about 4 different bacterial organisms. E. coli, Enterococcus faecalis, Staphylococcus epidermidis, and Staphylococcus aureus. You can see some of them are gram negative and some are gram positive. I didn't write it here, but Staphylococcus epidermidis and Staph aureus are both gram-positive cocci. They can both be found on the skin and so on. So there are 4 organisms that we're working with in our experiment today. But what I've done is I've taken 2 of them and I've mixed them together in what we'll call an unknown mix. And the goal of our experiment today is for you to try to determine which 2 of these organisms I combined together. And we're going to use special media to help us figure that out. So for the media that we're using for the exercise, we're using nutrient agar for the first part of the experiment. We've talked about nutrient agar before. It's a basal media. And then we're also going to use mannitol salt agar, specifically at 7.5% sodium chloride. And we're also going to use EMB agar. Remember, that's eosin methylene blue agar, if you remember that from last time. And that agar is also a selective media. So nutrient agar is a basal media. Remember, it will grow virtually anything as long as it is non-fastidious, as long as it doesn't have any very particular nutritional requirements. Mannitol salt is an agar that is selective and differential. It selects for 2 types of Staphylococcus, Staphylococcus aureus and Staphylococcus epidermidis, which just happen to be the 2 kinds of staph that are possibilities today. Now, MSA selects against virtually all other bacteria. Remember, salt tends to be very toxic to bacteria. There are a couple of Staphylococcus species that are exceptions to that. But remember, I also mentioned last time that it is possible for Enterococcus to grow on this medium, but it grows very poorly. It grows as very small colonies. And we do have an Enterococcus organism in our list of possibilities today. Now, MSA is also differential. It can differentiate different species and different strains of the same type of organism. And it specifically differentiates between organisms that can ferment mannitol and organisms that can't. So mannitol is just a sugar, it's just a carbohydrate. And it turns out that Staphylococcus aureus is able to ferment mannitol, while Staphylococcus epidermidis cannot. When Staphylococcus aureus ferments the mannitol sugar in this agar, the bacteria secretes an acid byproduct, and that acid turns the agar a yellow color. S. epidermidis cannot ferment mannitol, so it doesn't secrete any kind of acid byproduct, and the media stays its normal pink color. So both organisms can grow on MSA, but one of them is going to turn the agar yellow and one will not turn it anything other than pink. And that's what we can see on this slide. This is what a mannitol salt plate looks like before you put bacteria on it. You can see the agar is a light pink color. And that pink color comes from the indicator dye that's put into the agar. Remember, indicator dyes are going to be different colors under different pHs. On this slide, on this side of the slide, you're looking at a mannitol salt plate that has been streaked with 2 different organisms. Up at the top, you can see the streak lines here. You can see where the bacteria has grown. Up at the top here, we streaked Staphylococcus epidermidis, And down at the bottom, we streaked Staphylococcus aureus. And then we put the plate into the incubator for 24 hours. And when we took it out, it looked like this. So you can see both organisms grew. I can see something growing on these streak lines. So both types of Staphylococcus grew on the agar, but one kind left the media pink and the other kind turned it yellow. It's only Staph aureus that will turn it yellow. And it turns yellow because this organism secretes an acid when it ferments the mannitol sugar that's inside the agar. And that acid drops the pH and causes the dye to turn this yellow color. Now, the other selective media, or I should say the other special media that we're using is this EMB media. Now, this one's a little more complicated to understand, so make sure you jot down these special features of EMB in your notes. EMB is selective for E. coli and other fecal coliforms. Now, I have this term in quotes here because this is sort of a broad description of gram-negative bacteria that grow in the intestine and are often found in the feces. And we sort of call them very generally fecal coliforms. So E. coli and other bacteria that are gram-negative and grow in the intestine and are often found in the feces. Now, what it selects against, what it will prohibit from growing, is anything that's gram-positive. So any kind of a coccus, like Enterococcus or Streptococcus or Staphylococcus, those things are not going to grow on EMB. They can't grow on EMB. Now, like MSA, EMB is selective and differential. It can tell the difference between different species or strains of the same kind of organism. Now, MSA has mannitol in it as a sugar. EMB has two sugars in it. It has lactose and sucrose, two different carbohydrates. E. coli and other fecal coliforms, they can ferment these sugars, either one or the other. Now on this plate, if they ferment the sugars, what's going to happen is the colonies are going to turn purple. So this time it's not the agar that's changing color, it's the colonies themselves that are changing color. This agar is able to tell the difference between E. coli and its closely related cousins, the other intestinal gram-negative bacteria. E. coli will be purple, but it will also have a very distinct green metallic color. And you will especially see this where there are lots and lots of cells growing. Sometimes you'll also see, and I'll show you a picture of this, some of the purple colonies have this little dark area right in the middle. We call these fisheye colonies. I'll show you a picture in just a minute. But what's important to remember here is that E. coli, when it grows on EMB, it is fermenting the sugar, so it's turning purple, but it also has this strange green color. Only E. coli will show that green metallic color. Other gram-negative fecal organisms, other ones that can ferment these sugars, they're going to turn purple, but they won't have that green color associated with them. Now, there's one other thing to know about EMB. There are some gram-negative fecal organisms that cannot ferment lactose or sucrose. A couple of examples of those are Salmonella, which you've probably heard of, and another intestinal organism called Shigella. These can grow on EMB, but they can't ferment the sugar. So their colonies will not be purple. Their colonies will stay clear on the agar. And of course, there won't be any green color because only E. coli shows the green color. So this is a complex media. It can tell us a lot of information. Only gram negatives can grow on it, and they have to be intestinal organisms. But we can tell the difference between some of these intestinal organisms by how they grow, what color the colonies are, and whether there's any color changes associated with them. On this slide, you're looking at some EMB plates. Now, this plate right here in the upper left, this is what EMB looks like before you put any bacteria on it. It has a very unusual, dark, sort of purplish red color. This plate over here is what EMB looks like when you put E. coli on it. See this greenish color? It's very metallic looking. If you had this plate in your hand and you were able to tilt it back and forth under the light, you'd be able to see that the colonies are purple, but when you shine light on them, they sort of reflect off this green color. And it's especially clear where there's lots and lots of cells growing, like right here. Only E. coli will look like this on EMB. So it's very useful to us to know if we have E. coli in a particular sample. Now, on this plate, we can see other organisms growing on EMB. Take a look at this one. You can see this plate has been divided into 4 quadrants. This is EMB, even though it doesn't look quite as dark as this plate does. It is EMB, it's just under a strong light. So this is what E. coli looks like, again, when it grows on EMB. It's a purple colony, and again, this is just a big smear of cells. This isn't a colony, but it's a purple colored growth that shines this green metallic color under the light. Only E. coli will grow like this. There are other gram-negative fecal bacteria that will grow on this plate, though, and they'll grow like this. If they can ferment lactose or sucrose, they will be purple, but they won't have that green color associated with them. So any gram-negative intestinal or fecal bacteria that can ferment lactose or sucrose or both will be this sort of light purple lavender color when it grows on EMB. Down here, this is growth from an organism that is, again, gram-negative, intestinal, but it cannot ferment lactose or sucrose. And it grows as this sort of clear or colorless material. And finally, if we put anything on EMB that is gram-positive, it's not going to grow. Only gram-negative intestinal organisms can grow on this type of agar. So it's very helpful to us. So we have 2 types of agar today that are selective and differential. And if you go back and look at the possible organisms that are going to be in the mixed unknown culture, here's E. coli. Here's another fecal organism, but it's gram-positive. And then we have 2 organisms that we know can grow on MSA. So by streaking our 2 unknown organisms, we should be able to know what they are. I did want to show you this picture. This is an EMB plate. A quadrant streak has been done on it. You can see some individual colonies growing here. And again, there's that green color. This is E. coli. These colonies have a purple color to them, but they also have that fisheye, that dot in the center. I think you can just see it. All E. coli won't grow this way. Some does and some doesn't. But if you see it, if you see it on a plate, you know, again, you've got E. coli. The green color tells you it's E. coli. And some E. coli grow with that dot right in the middle of the colony. Very interesting. Okay, does anybody have any questions about these two types of culture media? Does anybody have any questions about what they select for and what they select against? Both of them will only allow certain bacteria to grow, and both of them will show different color patterns to tell us which species or which strain is growing on the plate, if it does grow. Any questions so far? All right, well, let's jump back in and look at the results we got. First, I want to ask the group a question, and you can type your answer into chat if you think you know. I told you a moment ago that in addition to the MSA and the EMB, we're also going to streak our organisms out on nutrient agar today. As part of the experiment, I took the 4 individual possible organisms and I streaked them out onto nutrient agar. Why do you think I did that? Why would that be part of my experiment? I'm trying to figure out which 2 of those 4 organisms are in our unknown mixed culture. So why bother putting those 4 organisms on nutrient agar first? I mean, that's not going to give me any information about the unknown mix. So Brooke's saying, is it a baseline? Am I looking for a baseline? What do you mean by baseline there, Brooke? What information am I getting from that nutrient agar plate? So all Emily's talking about whether or not we're restricting growth on some of the plates. We are on the special media. But what about that nutrient agar? Why am I bothering if the point of my experiment is to determine the organisms in the unknown culture? Why would I bother to take all 4 organisms and first grow them on nutrient agar? Remember, I'm going to choose 2 of those and I'm going to put them into a broth culture together, and then I'm going to put them on my special media. Why would I bother growing them first? It is a control group, Brooke. You're exactly right. What am I controlling? Like, what am I trying to show? Any ideas? You're all really close. Anytime we do an experiment that looks at bacterial cultures, like I'm going to use the bacterial cultures to do something. Anytime I'm going to do that, the very first thing I have to do is prove that those cultures are alive and that they can grow. If I have 4 cultures, I take them out of the incubator and I'm going to choose 2 of them and I'm going to mix them together. And then I'm going to spread them on some special media and see what kind. Well, that's great. But the very first thing I need to show is that they're alive and they're capable of growing. You can imagine that if I did the experiment without first using the nutrient agar, which will grow anything, if I did the experiment without doing that, and let's say I took my first culture and I put it on my special media and it didn't grow, how do I know it could grow? Maybe it was dead to start with. I have to show that I have viable cells before I begin. I have to do a control to show that all 4 of those cultures are alive and all 4 of them are capable of growing before I mix 2 of them together and keep going on the experiment. Remember, we're using living things. And so it's really important that before we make any big conclusions about those things, that we first demonstrate that they're alive and well. They're alive and they're capable of growing. Good. All right. So here's the procedure. First, I just drew 4 quadrants on the bottom of that nutrient agar plate, and using my best aseptic technique, I just made a very simple streak pattern in each quadrant with each of the 4 pure cultures. I didn't need to grow any individual isolated colonies or anything. I didn't need to count colonies. All I needed to do was make sure that I had viable cultures that were capable of growing. I put the nutrient agar plate into the incubator upside down at, again, about 35 degrees centigrade. That's a typical temperature for bacteria, for 24 hours. This is what the plate looked like when I took it out. Now I have 2 plates here. I didn't actually make these 2 plates. Students made these 2 plates. I just took pictures of them. But you can see I've got a nutrient agar plate here. You can just make out that the student drew lines across the bottom of the plate. And then the student took 1 culture at a time and just streaked cells onto the plate. All right, 1 from each culture, each of the 4 cultures. This student did the same thing. She just used a different streaking pattern. Hers wasn't quite as intense. But both students got the same results. All 4 of those cultures, the E. coli, the Enterococcus faecalis, the Staphylococcus aureus, and the Staphylococcus epidermidis, all 4 of those cultures were alive and well, and we were ready to go. So I chose 2 of those 4, and I mixed them together in 1 broth tube and put them in the incubator so that I could make some plates. I made a quadrant streak onto the MSA plate, and I made a quadrant streak onto the EMB plate. So I should get individual colonies growing if the organisms are able to grow on these particular types of agar. I put both plates upside down in the incubator for 24 hours, and then I pulled them out. So let's take a look. Here is the MSA plate. And we've got something growing on it. Now, I know it's hard to see from your angle, but I think you can make out there's an individual colony right there. There's another one next to it right there. There's an individual colony right there. There's another one right here. They're circular colonies. They have nice, even, smooth edges. So there is 1 organism growing on this plate. That's important to know. And of course, the agar has changed color, hasn't it? The agar has turned a yellow color where the organism is growing. Now this is something you'll want to jot down because this is obviously part of the results and it's important to us. I put 2 organisms in that broth tube. I streaked 2 organisms mixed together on this plate, but only 1 of them grew. I've got 1 colony type here. So only 1 of the 2 unknown organisms grew. And as it grew, it turned the agar yellow. Now this is the EMB plate. And your eyes don't deceive you. There is absolutely nothing growing. So I took that culture that had 2 organisms in it, and I made a quadrant streak on this plate, and neither one of those organisms was able to grow. This is information, right? This is information for me because I know what does grow on EMB, and I know what doesn't grow on EMB. So I have some data now. So let's walk through these results and see if we can figure out what two organisms I chose. First, let's ask ourselves about the first possibility, which was E. coli, if you remember. Could E. coli be one of the two bugs in that mixed culture? Remember, we got one organism to grow on MSA. We got nothing to grow on EMB. According to the MSA, could it be E. coli? Well, E. coli could be the one that didn't grow. That's possible, right? Remember what MSA will grow. MSA, if I go back here for us, MSA selects for Staph aureus and Staph epidermidis, and it will prevent virtually all other bacteria from growing. So if E. coli was in our unknown culture, it should not have grown on this plate. Maybe the organism that didn't grow was E. coli. Maybe, right? But what about this plate? This plate should have grown E. coli really well. E. coli grows great on EMB. Remember, it turns purple and it has that metallic green color. Nothing grew on this plate. So E. coli could not be one of our two unknown organisms. Does everybody see how I did that? It's a process of elimination there, right? If E. coli was one of the two unknowns, it should not have grown on the MSA because MSA won't grow E. coli, but it should have grown on the EMB. It should have grown really well on EMB. I should have seen purple colonies and they should have had a green metallic sheen to them. Nothing grew on that plate. So that's a piece of information for me. That tells me, oh, one of my unknown organisms, it can't be the E. coli. Does everybody see how that's done? Anybody not see how that's done? Sometimes we use our growth results in a sort of an affirmative way. Sometimes we can look and go, oh, look, we expected to grow, I'm going to make this up. We expected to grow a blue colony. And we did. Look, we grew a blue colony. But other times, we're not affirming with that. We're doing a process of elimination. Sometimes we say, well, if E. coli was in my water sample, it should have grown, and it didn't. So it must not be there. So we have good information so far that E. coli is not one of our unknowns. Now, let's look at the next one. Remember, the second organism that's a possibility is Enterococcus faecalis. Remember, Enterococcus faecalis is an intestinal organism, but it's gram positive. First things first, do we expect it to grow on MSA? No, right? There's only two things that will grow on MSA, Staphylococcus aureus and Staphylococcus epidermidis. So we don't expect it to grow on MSA. So once again, it's a possibility E. faecalis is in our mixed unknown culture because one of our organisms didn't grow on MSA. One of them did, and one of them didn't. So the one that didn't could be E. faecalis, right? What about this plate? What does this plate tell us? E. faecalis is an intestinal fecal organism, yes. And that's what we usually grow on EMB. But E. faecalis is gram positive, and only gram negative fecal organisms will grow on this media. So what do we think? Nothing's growing. Could E. faecalis have been in our culture? Well, yeah, right? We have two cases here of ruling out something with a lack of growth. Something in our mixed culture, something didn't grow on MSA and didn't grow on EMB. Could that something be E. faecalis? Yes, because E. faecalis shouldn't grow on either one of those plates. Could it be? Might be, right? We have to at least keep it in the running. We know it's not E. coli, but it could still be Enterococcus faecalis, so we have to keep that in mind. Let's look at the third organism. Could one of our unknowns have been Staphylococcus epidermidis? Well, we know Staph epidermidis grows well on MSA, and something grew on MSA here. But we also know that when Staphylococcus epidermidis grows on MSA, the agar stays pink. This is not Staphylococcus epidermidis, because whatever's growing here is turning the agar yellow. Does that make sense to everybody? We know that there are two kinds of staph that will grow on MSA, and we got something to grow on MSA, but we just don't know what. Could it have been epidermidis? No, it couldn't be. Because when epidermidis grows on MSA, epidermidis can't ferment mannitol. It just can't. It doesn't have the metabolic machinery to do that. So when it grows on MSA, the media stays pink. The agar stays pink. There's no acid byproducts made, no color change. So yes, it could grow on MSA, but the media should be pink, and this media is definitely yellow. So it's not Staph epidermidis, according to that plate. What about the EMB plate? Well, we don't expect staph to grow on EMB anyway. Remember, EMB will only grow gram-negative organisms, and Staphylococcus epidermidis is a gram-positive. So this plate, doesn't tell us much. It could be not growing on this plate, but this plate tells us it cannot be Staph epidermidis. So that leaves one more organism, which is Staphylococcus aureus. Could Staphylococcus aureus be in our unknown culture? We know Staphylococcus aureus grows well on MSA, and we know that when Staph aureus grows on MSA, it turns the media yellow. That's pretty good evidence right there that one of the organisms that I chose for that unknown culture was Staph aureus. Staph aureus is the only organism that will do this on MSA. It will grow because it doesn't mind all that salt, and it will ferment the mannitol in the media. It secretes an acid byproduct, and it changes the pH in the agar and turns it yellow. So that's a really nice sort of affirmative piece of evidence that Staph aureus was one of the two organisms. So we had Staph aureus as one of the two unknowns. What was the other one? E. coli, E. faecalis, or Staph epidermidis? What was the second unknown? Type it into chat if you know. If you need to see these again, let's look. We know Staph aureus was one of them, but there's another one of these bacteria in our unknown mixed culture. Is it E. coli? Is it fecalis? Or is it epidermidis? What do you think? I took two of those. I took two of them, put them in tube, streaked them out onto MSA, streaked them out onto EMB. We know one of them. We know one is S. aureus, has to be S. aureus. What's the other one? Brooks got it. Brooks got it. It's got to be Enterococcus faecalis. Enterococcus faecalis has to be the other organism by a process of elimination, right? By a process of elimination. I didn't actually grow any Enterococcus. So how do I know? Well, I know because I ruled out the other ones. It can't be E. coli because E. coli would have grown on EMB. And it can't be Staph epidermidis because Staph epidermidis would have grown on MSA and left the media pink. So the only one left is Enterococcus faecalis. You got it, Brooke. Good job. So this experiment is a little unnatural in that we started off knowing the possibilities, right? We started off knowing 4 organisms. And then we had this unknown. And we knew that it had 2 of those 4 in it. So we had a lot of information just to begin. A lot of times in the microbiology lab, we don't have any starting information, right? We have patient symptoms. We have signs that the physician has recorded. But a lot of times when a urine sample arrives in the lab or a blood sample or maybe some material from an abscess or some sputum from somebody who has pneumonia or bronchitis, we get that material in and the physician says, hey, what is this? What bacteria is growing in here? We don't know anything else besides the signs and symptoms. So we have to use a lot of special media to figure that out. First of all, we have to have some basic knowledge about what is likely to be in a urine infected bladder, what is likely to be growing in a stomach of a person with ulcers, what is likely to be growing in the lungs of a person with pneumonia. We have to have some understanding of what we would expect, but it's usually a pretty long list of possibilities. So that's why we have to do a lot of testing. We gram stain it, and then we grow it on these special media. And remember, there are lots of special media, not just MSA and EMB. There's lots of them. So we make lots of these plates and we look at the results and we gather data and we eliminate possibilities one by one based on what we find. All right. Ours was just an experiment. We knew it had to be 2 of these organisms. So we had a pretty short list of possibilities, but still we had to read those plates and we had to know what grows on EMB, what doesn't grow on EMB, what grows on MSA, what doesn't grow on MSA. And by knowing that and looking at the actual plates, we can narrow down our possibilities. All right, good. Good job. That's it for special media. When we get together next time, we're going to talk about some other media which we use to help us learn things about unknown bacteria, to help us diagnose or determine what kind of bacteria are growing inside of a patient or out in an environmental location. And that is by looking at oxygen requirements. We haven't talked about metabolism yet, and we haven't talked specifically about oxygen pathways and such. But we're going to take a look at the media that's available to us to determine what kind of oxygen requirements an organism has. We have the ability to set up culture media that has oxygen in it, that doesn't have any oxygen in it, and that has everything in between, has small amounts of oxygen in it, or maybe a little bit more or a little bit less. We can set up media with different amounts of oxygen, and then we can look to see if a bacterium can grow under those conditions. If they can, then we know that they need oxygen, or that oxygen doesn't bother them. If they can't grow in the presence of oxygen, that's an important piece of data for us, because different bacteria are going to have different needs regarding oxygen. There are lots of bacteria that find oxygen toxic. They simply cannot grow in the presence of oxygen. A good example of an organism that finds oxygen toxic is the organism that causes tetanus in human beings. Now, this organism has the name Clostridium tetani, and it causes this disease that we know of as tetanus, or some people refer to it as lockjaw. You and I are vaccinated against this tetanus bacterium because tetanus kills people, or at least it used to. Thank goodness there isn't much tetanus around anymore. But Clostridium tetani is a bacterium that finds oxygen toxic. It doesn't need it, and it finds it toxic. So if it gets exposed to oxygen, it can't grow. In fact, it dies. And if you think about how people get tetanus, it makes sense, right? Most people get tetanus when they get a puncture wound, a deep puncture wound. Like, you probably have heard stories about people stepping on rusty nails or accidentally stepping on something sharp. And if there's any Clostridium tetani on that sharp pointed end, it'll get jabbed down deep into the tissue. And down there, there's not as much oxygen as there is out here in the air. And so the Clostridium can grow deep down into our tissue beds. And that's how tetanus takes off in a person. So some organisms, they absolutely require oxygen, just like you and I do. Some organisms, they find oxygen toxic. And there are still other organisms that they need oxygen, but they need it at low levels. Or maybe they can use oxygen, but they don't have to use oxygen. They can switch back and forth. There are all kinds of bacteria with regards to oxygen needs. Now, here's a question. You can go ahead and type this right into chat if you know the answer. What exactly, let's think about ourselves for a minute. Let's leave the bacteria aside. You and I need oxygen. We have an absolute requirement for oxygen. If we stop getting oxygen, like let's say we stop breathing, we don't have long to live, do we? We've got a few minutes to live without oxygen. What exactly are our cells doing with that oxygen? Why do we need oxygen? What is the oxygen for? Does anybody know? People who have a heart attack, who fall down unconscious, their heart has stopped beating, they are not breathing. That's a medical emergency, right? And when the EMTs arrive or the other medical staff gets to work on them, what's the first thing they do? You clear the airway, right? And you start breathing for them, or you put oxygen on their face, right? It's critical. They don't have long to live if they don't get oxygen down into their system, down into their body, and into their blood and to their cells. The question is, why? What do your cells do with that oxygen? So Brooke is asking to carry nutrients or gas exchange. Well, the red blood cells are carrying the oxygen, right, Brooke? When you breathe it in from your lungs, from those alveoli in your lungs, you exchange oxygen and carbon dioxide in the red blood cell. The red blood cell lets go of the carbon dioxide so you can exhale it. And the red blood cells take in the oxygen. And then your blood carries those red blood cells all the way through your body. And the red blood cells give the oxygen to your cells. And your cells take that oxygen in and they use it. But the question is, what are they doing with it? What do your cells do with oxygen? As humans, we have to have it or we will die. There's no substitute for it. So again, we're going to talk about this more in lecture material when we get into our metabolism lectures, but it's all about, yeah, Rue has it. It's all about ATP. It's all about ATP. Everything is about ATP in cells. Remember, ATP, or adenosine triphosphate, ATP is this little molecule that acts like energy currency in the cell. When the cell needs some energy to do a chemical reaction, it gets it from ATP. So, in order for your cells to make ATP, they do a long, complicated process called cellular respiration. It starts with glucose, a molecule of glucose, and it ends way down the line with ATP. And it all happens in your mitochondria. The glucose gets broken down, and all of the electrons that are in those covalent bonds, remember, all a covalent bond is a pair of electrons that are being shared between two atoms. So when that bond is broken, the electrons get released, and electrons carry energy with them. So the glucose molecule that feeds your cells, it gets broken apart, and all of the electrons in the glucose get released. And those electrons get harvested in this very special structure in your mitochondria called the electron transport chain. And what that chain does is it harvests all of this electron energy and it uses it to make ATP. But here's a point that a lot of people don't know or don't think about very much because, it's all about the ATP, right? At the end of the electron transport chain, those electrons have to go somewhere. Nature doesn't like electrons flying around unsupervised. Nature wants electrons to be in bonds. And remember, nature likes valence shells around atoms to be full. Paired electrons are the most stable structure that there is in a molecule. So we've taken all these electrons out of glucose and we've passed them down this structure called an electron transport chain, and we've taken their energy And we've used that energy to make ATP. Awesome. But now we're left with all the electrons. What do we do with them? We give them to oxygen. Oxygen is called the final electron acceptor at the very bottom of the electron transport chain. The final electron acceptor. Remember, oxygen has a very high electronegativity. It draws electrons to it, right? So down at the bottom of the electron transport chain, after the electrons have had their energy withdrawn, essentially, they bind with the oxygen and a little bit of hydrogen, and water is created. Nature will not leave those electrons floating around. They're going to be taken by an atom or into a molecule. And it turns out that for us, that molecule is oxygen gas. We can't go without it, because if we don't have oxygen, then there's nowhere for the electrons to go. And the electron transport chain will just stop. Our mitochondria will just hit the brakes. No more ATP will be made. And the truth about ATP is we don't store a lot of it in our cells. We make it as we need it. So we need to be breathing fresh oxygen 24 hours a day, 7 days a week for the entirety of our lives, so that every cell in our body has a fresh supply of oxygen so they can make a fresh supply of ATP. If you run out of ATP, you're dead. There is no life without ATP. And here's a really fascinating fact. Every single living cell on this planet needs ATP. They've got to have it or they won't survive. Bacteria, some of them are just like us. Some of them need oxygen just like us. And they do the exact same thing that we do with it. Although they don't have mitochondria, they still make ATP and they use oxygen as the final electron acceptor. But other bacteria have evolved to use a different molecule as their final electron acceptor. Some bacteria use a molecule called nitrate, and some bacteria use a molecule called sulfate, and some bacteria use a molecule called carbonate. They don't need oxygen. They don't need it. They just need their final electron acceptor. And as long as they have it, they can make ATP and they can stay alive. So it's all about ATP. I say that a lot with students. Everything comes down to ATP in a cell. But bacteria are very diverse in how they, what molecule they use at the bottom of their electron transport chain in order to make ATP. So, a little bit of metabolism today. That is coming up in lecture when we talk about metabolism. And next week in lab, we'll do some testing. We'll show how different organisms can be grown in the lab with regards to whether or not they need oxygen. And if they do, how much do they need? That'll be coming up next week. Any questions? Any questions about anything we talked about today or when we were together earlier in the week? Remember, you have some homework questions, some lab homework questions to answer about this week's material. Those are due Sunday. And if there's no other questions, I'm going to let you go. Okay? Enjoy the rest of your Friday. It's a beautiful day out today. I hope you have a nice weekend. Stay safe. I'll see you next week.
Wait, so the lab questions that you're talking about, you said they're due Sunday?
Yes, as always, lab homework questions due Sunday by midnight.
I thought I already did that. I thought it was due tomorrow.
Saturday?
I thought
We don't
I-
usually have things due on Saturdays. We have quizzes that are due Wednesdays and Fridays each week, and then we have lab homeworks that are due on Sundays.
Oh, yeah, I see. I think I am confusing that with the lecture quiz.
Oh yeah, sure. Yep.
That is due today, I think.
Yeah, by midnight tonight.
Yeah, I already did it, but I think what I'm just gonna do is just do stuff before the due date, so
Oh,
I'm not
sure.
being track of it.
That's
So,
always fine. You can always submit things early.
mhm.
It's not a problem at all.
Okay, so then for week 6, right, which is next week,
Yep.
you already posted the videos for the lecture quiz?
Yep.
Okay, all right, I'll get on that.
All right, very good.
Thank you.
All right, we'll see you next time.
See ya. Bye.